Good morning, everyone. My name's Joe Avellone. I'm the president of the Carter Foundation for Neurologic Research. I want to thank our host, Ate, who has run the program, the Kids with SPG3A website, so beautifully over the last several years. And with me today is Dr. Craig Blackstone, who's a professor of neurology at Harvard Medical School, and also our senior scientific advisor for the foundation that is helping us guide the research into SPG3A. If we go to the next slide, please. Just wanted to talk about what we wanted to do in our update today. And typically we would always start with the mission. As you remember, our mission is to really look for therapies that are geared towards hereditary spastic paraplegia generally, but specifically, at least initially, SPG3A and indirectly SPG4 because there are some relationships together. We are trying to do something that takes us from the basic science, where there's a more of an understanding of the biochemistry of what is causing these diseases, and move it forward to an animal model eventually that shows the neurologic problems that these children have, and then more importantly, how our potential therapies can improve their condition in the model. If you can have a reproducible animal model that can do that, then you can go ahead and develop drugs either through institutes or biopharma companies, et cetera. And that's really what we're trying to do with the Carter Foundation. So if you go to the next slide, please. We have a series of projects that we've been running now for several years. And the first one is based on a human neuron in a petri dish, essentially, using stem cells that have been promoted to become human neurons with the exact mutation of an SPG3A condition. And we've been testing various drug hypotheses on that for the last several years. And I'm going to turn it over to Dr. Blackstone now to ask if he can tell us what we've been learning and where we're going from here with those projects.
Yeah, sure, Joe. Thanks for having me. I can certainly start out by talking about the bigger picture of where the HSPs are. And as many of you know, over many years, a lot of genes were identified, and that was sort of the first clue as to what might be causing these diseases. And of course, genes make proteins, and by studying those proteins, we get ideas on how we can potentially treat these diseases. But as you mentioned, translational science requires us to take those insights and translate them into actual therapies. And to do that, we have to move beyond just understanding the individual protein and understand how it works in the context of individual cells or organisms. And ultimately, of course, that gets us closer and closer to what we need to do to treat patients. So this project you have mentioned here is from Dr. Lee at University of Illinois School of Medicine. And what she's trying to do is to try various hypotheses based on the genes that we know. In this case, it's on lipid metabolism or lipid modulation within the cell. And lipids are very key building blocks of cells, and they have to be distributed in different parts of the cell. And of course, a very big cell like a neuron, has challenges in that, and that might be related to this disease. So when we think about how to study this, we want to study it in relevant systems. And one relevant system that's just been really advanced over the last few years is iPSCs. And iPSCs are induced pluripotent stem cells. So they're cells that are made from skin of patients, and they're changed into stem cells, which can then be differentiated into neurons. So it allows you to study a sort of a disease mechanism or a response to therapy in actual cells from patients themselves. So I think that this is one very good model system. Of course, it's not an intact brain, but it's a key part of the brain, the neurons and the glia that are important in this disease. So what she's doing here is using these cell lines and trying various therapies that are based on the understanding of the genes and seeing if it restores normal function to these cells. So when she's looked at these cells in the past, she's noted some abnormalities compared to cells from a patient who does, or from a person who doesn't have a disease. And the goal is, of course, is to make the abnormalities go back to looking like the cells of a person without any problems. And that's what she's been doing. So that's one way to test these therapeutic agents, is to see exactly how how they respond or how they affect those processes within the cell. Now, of course, that's only one part of this. We're not sure if those abnormalities are related to the ultimate disease, but it's a starting point and it's just one model system. And as we'll talk about, we use multiple different model systems because, only people have HSP. All mice and flies and cells, they can mimic certain parts of the disease and we can learn from that. But these cells don't have HSP, only people do. So here she's using a couple different pathways. One is called BMP signaling, as I mentioned, the other is called lipid modulation, and there's several others that we're thinking of trying. The advantage of doing it in cells is it's fast. We can get early indication of whether it's working, and that might allow us to prioritize which agents we use for more complex systems like mice or flies. And of
Yes.
course, I think that that's exactly what you have in your bottom point here is that these types of studies will inform our mouse model studies, which of course, as we know, are beginning now.
And I guess if I could just add, Dr. Blackstone, that just in the last 6 months, I would say that the data has been somewhat encouraging from her studies around lipid modulators, so that there is some activity that seems to impact beneficially the HSP cells when these lipid modulators are used. So that's allowed us to believe that we can use this now in the animal model, which we'll talk about later, and obviously as a hypothesis that could help. And we're going to now starting just as we speak, looking at this other pathway, once again in the stem cell model, what's called the BMP signaling pathway, which is looking at a different kind of thing that we believe could beneficially impact the neurons. So this is kind of the basic building blocks of getting to the animal model, showing that it allows us to use it in these kind of longer term, harder to do models down the road. If you want to go to the next slide. And so another one of the more complex models then is the fruit fly, which the formal name is Drosophila. And as you might remember, we've been running a couple of projects through Dr. Nemo Sullivan at the University College Dublin. And actually, it's led to some interesting results initially that lead us to a hypothesis about how to move from here. So without being mysterious about it, I wonder if I could ask Dr. Blackstone to explain what the model is and what we have learned so far.
Sure, Joe. So again, when we're talking about children with SPG3A and SPG4, there's a subset of people, and these are two very common HSPs, but there's a subset of people who have a more severe form, and they have in general, specific types of mutations. Now, SPG3A is the one we're focusing on here, and almost all the mutations that are known across all patients with this disorders are what they call missense mutations, which means it's a change of one amino acid to another within the protein, and that's sufficient to give disease. However, there's a subset of these children that have a more severe form, and they tend to have a specific type or specific location of these changes. So one of our hypotheses in studying the flies is, can we see differences with these severe forms versus the more common, less severe forms? And does that teach us something about the disease mechanism that could be important for how we treat these? So when you look at this slide, you see that there's this elastin M38T, and that's methionine to threonine. So that's a specific change. In the patients, the number is 408. And that's just because the numbering and the size of the proteins is different from a fruit fly to a person. But it's essentially the same change mimicked in the fly as we see in the children with more severe forms. And you'll see in the next thing, one of the hypotheses is that you have we all know that we have one, for most of our proteins, we have one copy from our parents, one from our mom, one from our dad. And in these diseases are what are called dominant, which means only one of the proteins is abnormal, the other one's normal. So the hope is that, well, if we can get rid of the bad one, maybe the normal one, maybe that's enough for the cell to do just fine. And we think that's true for SPG3A in particular, and that's the one we're talking about. So one idea is, can we just remove that bad elastin? And as Joe has listed here, that's called a toxic gain of function. There's this protein is doing something abnormally that's causing a problem. And if we can remove it, that the patients might get better. And one way to test that hypothesis is to make mutations in cell lines or in organisms. One reason we're doing it in flies is that it's fast. We can do this similar studies in mice and other species. However, it's much slower. It takes a lot longer to make these mutations and test them. So an advantage of using the flies is it's very fast. And just like the cell lines that we described earlier, it can allow us to do more definitive studies in mice, which are far more expensive and take far longer. So it's really pretty simple. We want to target that bad elastin, we'll call it, and not damage the good one. And there's ways to do that using ASO approaches, and that's using specific RNA strategies to remove the bad one, and it's called antisense oligonucleotide. And this is used in several diseases, to treat them with patients in a variety of disorders, most notably spinal muscular atrophy, also some forms of ALS, and it's been proven to be very effective and safe. So it's a very promising direction for specific types of mutations. Now, again, some of these has to be customized to one patient or another, depending upon the approach we use. But what we're just trying to do here is to see if it's viable. What if we take this and just remove the bad form? Now, what's turned out that's been very useful about the fruit fly model is, in fact, it has been shown that, at least in the preliminary studies that Neve has done, that the form that mimics the more severe form in the humans is also more severe in the flies. Now, that doesn't necessarily change the therapy that we would give. There's a chance that the same therapy would work for everybody, but it does mimic an important feature that we see in people, and that could be very helpful for us to understand why some children get a more severe form than others, and whether that impacts the way we would approach them in terms of treatment. But I think the particularly encouraging thing about the ASO approach is that I think it would work for not only the severe form, but the less severe forms and across a wide variety of mutations. So if we can show conceptually that this works, I think it will open up a therapeutic option for a large number of people with SPG3A. Now, SPG4, which we're not talking about as much here, is a little more challenging because some of the patients with that have what they call deletions. So one of their genes is gone and they still get the disease. So it's not as clear with SPG4 if it's safe to remove the bad form, that maybe you still don't have enough of that protein. So potentially we would move in a different direction on SPG4. But I think for SPG3A, this ASO approach of removing the abnormal protein is a very promising one.
Thank you, Craig. And I would just add that the so-called ASO approach, as he mentioned at the very beginning, has been used now with some other diseases. So it has been, there are approved therapies that for some very rare diseases have been shown to benefit. And this is very important because it means that the model theoretically it can be done in humans, obviously very carefully. And each one is very individualized, but it is an approach to therapy that has some promise for us. If you move on to the next slide, we now get to the mouse. So once again, I said at the beginning that we're trying to bridge the gap between understanding things at the biochemical cellular level and getting them to a human model in which we can test therapies. And the best way to do that for these kinds of neurologic diseases is a mouse model. We started working with Jackson Laboratories about probably 9 months ago now. It takes a long time to kind of build the experiments and create the mice with the right cohort numbers and with the right mutations in order to do these things, but we're there. So we have now been working with them about 9 months. We have created what's called a human mutant mouse model. And we are ready now, in fact, are actually starting the first of the experiments, which is the lipid modulator that we learned about from the stem cells, and see if we can show an improvement in these mutant mice in the next several months. But Craig, if I could ask you to just back up and talk a little bit about what a human mutant mouse model is, and then how it's going to apply to our experiments.
Yeah, sure, Joe. So yeah, as we've talked about, in terms of doing translational science, in terms of taking the insights we get in cells all the way to actual therapies for people, there are some important steps along the way. We need to show that it's working in human cells, and we're doing that. But also, historically, it's been very important to show it in a larger animal model, like a rodent versus a fly, and to show that some key aspects of therapy, specifically whether you're engaging the target that you're trying to hit and whether you see an improvement in the symptoms, are very critical toward moving it through the regulatory process and making sure that what we give to patients is effective and safe. And the mouse model allows us to do both of those things. When we talk about a humanized model, it means in the area around the mutation that that entire piece of DNA is from a human. And you'd think, well, how can you do that? Mice and humans are so different. Will it behave the same? And it turns out that actually this protein is very similar between mice and humans. So it's safe for us to put the human sequence in there. Now, why would we do that? Well, if we're going to start using therapies based on specific sequences that are present in humans, we want to have that sequence present in our model systems. So in other words, even though the sequence is similar in a mouse to a human, it's not identical. But if we put the human piece into the mouse gene, just a portion of it, it allows us to test specific therapies that are targeted to the human DNA sequence. And that's what the humanized model is. Now, another thing we've done with this model is we've paired it with another HSP gene that interacts with this protein to make the model more severe. And if you think about it, when you have a small animal like a mouse, and you're thinking about a big organism like a human, and the neurons in the human are 1 meter long, and the mouse is only about 10 centimeters long. So it's very hard to mimic a disorder like HSP, which is a disease of long neurons in a small animal. And one way we can do that is by sort of making another mutation and making a more severe form in the mouse so that we can do this quicker. And one thing that we've been able to do with this particular model that we're working on with Jackson Labs is the mouse develops its symptoms very early, in early adulthood. And that allows us to test therapies without having to wait 1.5 years or 2 years, through the mouse lifespan. We're able to test it very, very early. And that's been very effective so far in some of our preliminary studies. So as Joe mentioned, we have the option to test multiple different pathways, not only the sort of cellular mechanisms like lipids or BMP signaling, but also some of the gene editing and oligonucleotide therapies that we talked about. Now, I can mention one thing about gene editing is that's listed on this slide, and that's called base editing. That's actually going in and changing the abnormality in the gene back to normal. So that has some very attractive features. It's a one-time thing, and you've created the normal gene. The challenge with some of these therapies also is getting it to all the cells that it's needed to get to in the brain. And that's where an organism like mouse is helpful, because we have the same issue. How do we get it to all the right cells? Whereas if we're studying a cell in a dish, anything we do will get into the cell. The accessibility is very easy. However, it's not so easy in an animal like a mouse. So that is closer to what this human situation is going to be. So we not only have the advantage that this mouse has the human sequence, but it has some of the same challenges that we have in terms of delivering therapies to humans, is we have to get it to the cells that need it. And this sort of gives us a little early indication of whether that will be effective or not. As Joe said, these take time, and we've been able to sort of prioritize our therapies based on what we've learned in those other systems that we mentioned, whether it's cell lines or flies that we can do quickly. We still ultimately want to move closer and closer to humans, and that's what this model allows us to do, both for gene-based therapies as well as for the typical small molecule therapies that can ultimately give rise to traditional therapeutics like medications that you take by mouth. And as Joe mentioned, one of the things that in this step that we can do is if we do get promising results in our mouse models, then we're able to start moving forward in terms of discussing with pharmaceutical companies or larger institutes, moving it more toward human studies. And those, of course, are far more expensive, a lot of regulatory burden. So it's very important that we have as much data in hand when we go to that next step, to ensure that we can continue to move forward in that process, because that's what those firms and institutes will expect. They will expect very strong data supporting the therapeutic ideas that we have.
Thank you. So this use of the mouse model now, especially in these four ways that we're thinking about, is really the culmination of all the work that we've been doing for the last couple of years with these other more basic science-related experiments. So it has been a continuation, and now we're at a point, especially with the mouse model developed, that we can start. In fact, the first experiment has started, and it is on the lipid modulator. And of course, getting the dose right, getting the right lipid modulator, there's lots still to learn in science. But there's been a lot of thinking that's gone into it too. So we expect to have our first early results on whether the lipid modulator theory is working out in the mouse model within several months. And certainly by the next time we have an update. And also, we are now evaluating what kind of a BMP signaling pathway type drugs we could also use. And then, of course, there are the more complicated ASO and or gene editing. So our effort is to cascade these over the next period of time. Let me put it that way. Lipid modulators and BMP signalers are known, let's call them small molecules that have been, in many cases, used before, certainly in animal models, if not in humans. And wouldn't that be wonderful if we can have a safe drug versus trying to manipulate DNA or RNA as the other more advanced therapies are, although the other more advanced therapies may be much more effective. So everything is a trade-off, but we are making significant progress. If I had to sum it up, I guess if we go to the last page, I would say that we have been making good progress over the first two phases of our research. We haven't had any evidence that any of these hypotheses don't work, and we've had some evidence that some of them do work. And I think that's a very important thing in science. That's progress. And I would say we can be sort of, I would say guardedly optimistic. Guardedly because science can go in a lot of different directions, but optimistic because so far these hypotheses have been playing out in the models as we get to the more complicated ones. Think that we will have more information probably within the 6-month time frame. But I do think that there's a lot of good momentum behind what we've been doing so far. And I'm very pleased, especially pleased with the leadership of Dr. Blackstone. A lot of these ideas really were developed by him during his years at the National Institute of Health and then later refined since he's been at Mass General. But they are playing out, and we're very pleased with the progress so far. Dr. Blackstone, any final words?
Yeah, I just mentioned, Joe, again, as you said, and that it really is important to pursue multiple different paths to therapeutics because there's things that can go wrong. I think that one of the things that we're increasingly thinking about is that we will want to ultimately treat patients with more than one, that it's not going to be an either or. Like even if we treat with these ASO therapies, we're probably still going to want to treat with medications. We're going to want to sort of hit the abnormalities in multiple different ways because no one therapy will probably be completely effective. And we're hoping to get synergy with different types of therapies. And one of the really encouraging things, as you brought up about some of the pathways, is a lot of these agents have made it at least partway through the development process in terms of drugs. So there's a lot of information. In many cases, the safety has already been assessed, and that allows us to move things forward quicker than if we had to start from a brand new molecule and go through the entire drug development process. Many of the compounds were tested have gotten a fair distance, and some of them have gotten very far along the development process for other disorders often. So a lot of the hard work in many ways and some of the safety work has already been done. And that allows us potentially to move forward quicker. And that's, of course, the traditional drug pathway. I would emphasize that for the RNAi and the gene editing, well, especially the RNAi, the ASO therapies, those can move quicker to human studies than the traditional drugs. So our hope is that, that we will be able to move some of these further along toward treating people with these disorders, quicker, not only because the ASOs can go into humans faster because there's been a lot of safety studies done to show that that's a very viable approach, but also the fact that our small molecule therapies have also been tested extensively in either preclinical models or in many cases in humans, I think will allow us to shorten the time to taking these ideas to people with these disorders.
Thank you. And time is the word. We all understand that these children, every day is another day in their life, and we want to advance these projects and hopefully these treatments as absolutely as fast as we can. And our commitment is to continue to do that. I want to thank, first of all, Dr. Blackstone for all his time and effort and really his leadership in advising our foundation. And I also want to thank our host, Ache, and her husband, Juan, for the wonderful website that they maintain that allows us to communicate with all of you. Thank you very much.
Thank you.
Thank you.